Okay, hi everyone. So now we are going to continue on the postmortem toxicology. All right, so what is postmortem toxicology? So postmortem toxicology is a discipline that is used to determine whether the alcohol, the drugs, or the other poisons that can cause or contributed to the death of a person. So whenever that we are talking about postmortem toxicology, postmortem is death lah kan. Okay, so basically it is the discipline whether we want to determine whether the alcohols or the drugs or the poisons may have may have caused or may have contributed to the death of the person. Alright? So usually, for postmortem toxicology, it is usually in the cases of suspected drug intoxication cases, yeah, in terms of homicides, in terms of arson fire death. Okay, so we want to make sure that whether the person had died from arson or from any other substances. So we need to be able to deduce those things lah. Alright. Motor vehicle fatalities, and lastly is whether the death is due to the natural or unnatural cases. Right? Alright. So, moving on. Postmortem toxicology, usually the common analytical methods in terms of postmortem toxicology will be using such as immunoassay, chromatography in terms of whenever that we have the sample, we suspected that there is a poison inside the sample that the evidence that we've got. So we need to be able to separate those compounds inside the sample to be able to determine the identity and the quantity of it. All right, so because we want to know if this poison, for example if this poison has caused the death of the person, we want to know the dose, we want to know the concentration. So all of these analytical methods might be useful for those things, which sometimes might require certain modifications lah, because usually we would have some sort of like a general standard method, but sometimes you need to have modifications through the method due to the differences in your sample matrices, right? Therefore, we need to be able to give acceptable results with whatever compounds that we have, whatever samples that we have. Okay, so that's why we would have some sort of like a modifications to it. So if you remember previous slides that we have looked at, toxicology, or specifically forensic toxicology, is one of the expensive side in forensic lah. It's just that because we need to be able to develop a method and validate the method throughout uh, as time goes by. Okay, so that we need to keep on updating our method because of the evolving of the drugs and poisons that are available. Okay, so that's why we would have some certain modifications to it. All right. Moving on. Okay, so this is an example of postmortem toxicology results in terms of the form. All right. Basically, you can have a look at this. Okay. For example, what kind of sample that we've got, whether the apa the person have a history of alcohol abuse, have a history of substance abuse. Okay, so all of these are important in terms of where we want to apa to deduce whether he or she has been poisoned or have been all of these things have caused the death. All right. So the specimens, for example, now you would see that the specimens here, there are urines, vitreous, bile, liver, stomach content, and blood. All right. For the blood, you need to specify the collection site. All right, because the blood it will be available throughout your body, so you need to be able to specify which part of the body that you've got the blood. All right. So now, based on this form, we are going to look into each of the specimen item. All right. So first of all, first specimen type, which is blood. Okay, blood. In living patient, the drug the dose of the drug is mostly correlated with its concentration in the blood or the plasma. So usually the concentration of the drug we will measure from the blood or the plasma. Okay, so the blood has therefore been used as the primary specimen in post-mortem toxicology. All right, so when we say that what is the primary specimen in terms of forensic toxicology, then it will be blood if it is available. Okay, so first of all, you must go to blood. Okay, if the body has been decomposed and there is no more blood, then we would go to another route. But usually, blood is going to be the first evidence or the first specimen that you need to collect. All right. In most cases, postmortem blood is relatively fluid and typically has a numerous small clots. All right. So usually, the postmortem blood, blood is fluid, but sometimes you will have some clots uh, postmortem. All right. So the sampling can be used by a syringe and a large gauge needle. Okay. All right. Postmortem blood concentration of many drugs may vary from site to site due to the process known as post-mortem redistribution. All right. As a result, much attention has been focused on the site of the collection of the post-mortem blood samples. All right. So based on this, you need to be able to select which part that you need to collect the blood from. All right. So for example, if I might say that, okay, for example, if you did collect the blood near the liver, so you would expect that it will be the poison, for example, the poison would have a much more concentration, much higher concentration near the liver where it the liver is where the poison has been metabolized. And you would also would indicate that the blood or the poison might be in its metabolite form. Okay, because it has been metabolized by the liver. So due to this, you need to indicate where you did get the blood from. For example, if you got it from the heart, then you need to be able to see, you need to be able to deduce whether the blood might consist of the poison or the metabolites of it. Okay, so you need to be able to know these things. Like I need to be able to put the specific part where did you get the blood from. Okay. Postmortem blood specimen from two sites, the heart and the peripheral, should be collected at every autopsy. So basically, it has been indicated that you need to get the blood from two sites, which is the heart and the peripheral. If no autopsy is performed, then only peripheral blood should be collected. Alright. Peripheral blood specimens are usually obtained from the femoral vein. Okay, leg veins are preferred due to the anatomically presence of a larger number of valves that resist blood movement from the intestine. Okay, so usually we will get the peripheral blood from the leg because the veins are much more larger there. All right. The peripheral blood, you should be able to take about 10 to 20 ml using a hypodermic syringe. If possible, up to 10 ml of the peripheral blood should be collected. Right? So, in terms of the blood specimen, the more or less 10 to 20 ml of blood that you need to collect. Do not milk the leg in order to increase the specimen volume. All right? So, this is just some precautions lah, right? The source of the peripheral blood specimen should be noted on the specimen container. So you need, as I mentioned before, even in the form itself, you need to be able to say, or you need to be able to indicate where is the blood come from, whether it's from the peripheral blood, for in this case leg veins, whether it's the right leg or left leg, right? So you need to be able to indicate these things. All right. All right, so this is just a figure showing the veins of the leg. All right, so this is the common femoral vein. So this is the deep femoral vein. So basically, you can see that this near this leg, the femoral vein is quite large. All right, so it is much more easier for you to get the blood from these veins lah. Right? Okay. All right. The second type is urine. So urine collected at autopsy provides the toxicologists with qualitative anti-mortem drug exposure information. Okay, as you know that when the drug has been metabolized by the liver, and then it will be excreted by the body through the kidney into the urine. Okay, so now you need to be able to know that the drug may not be, or the drug or the poison may not be in its original form. It's usually in terms of metabolites. So even though the presence of metabolites, you are still able to say that, oh, the metabolites of, for example, paraquat is present inside the urine specimen. So you'll be able to deduce that this person has been ingested or has been administered with paraquat. Alright? So the accumulation of drugs and their metabolites in urine results in a relatively high drug concentration, facilitating detection of an exposure to a potential poison. So that's what I've mentioned before. Usually the drugs or the poison may appear as a whole, but sometimes it will appear as a metabolite. So so now you need to be able to understand that the drug might not be excreted as the drug as a whole drug, but sometimes it will appear as a metabolite. All right. So positive identification of drugs in urine indicates past drug use, but does not indicate when or how much drug was ingested. So that's what we have said earlier, that urine usually we will do it as a qualitative drug exposure information lah. So because excretion is the end part of it. So usually we would say that, oh, this person has been ingested this drug prior to the testing. Okay, but we don't know how much is it. We only know that it is present inside his or her system. Okay. All right. The third specimen is bile. Okay, so bile is something near your liver, kan? I cannot see. Okay, yeah. So specimen type bile usually is something in your gallbladder. All right. So it is in your liver. So many drugs and drug metabolites accumulate in the bile. Okay. So from these ones, you need to be able to. Okay. The qualitative finding of the presence of drug or metabolites in this specimen is important for the documentation of historic exposure to specific agent and chronic drug use history. So, from bile, okay, it is usually being indicated that the drug has been, or the poison has been used way before the time of testing, as what we expected in urine. Okay, we can have a qualitative analysis of it. Okay, from the bile specimen, we have detected that there is a drug presence and its metabolites. Okay, so we can deduce that this person has been exposed to the drug way before the time of testing, because there is no way for us to know that how much he or she has been ingested or administered the drug or the poison. But for some reason, as it is one of the site that this drug has been accumulated, then we can have some sort like a documentation of the historic exposure. Okay. In the absence of urine, bile has been also been useful in terms of the alternative specimen for alcohol analysis. Alright? Historically, bile has most been often been used in the determination of opiates in general and morphine in particular. So, because the use of these opiates and morphine for some reason that uh, the the person would not be, apa orang kata tu, dia mesti dah ingest benda ni lama. So the drug has been accumulated in his bile. Okay, so that's why we use the bile as a specimen in terms of for these opiates in general and morphine in particular. So straight away when we say that, okay, I want to test about for morphine, so you straight away, okay, we can use bile, but it is just a qualitative analysis. You would not know in terms of how much he or she has been ingested or administered the drug. Okay. I think this is the fourth one. Vitreous humor. Okay, vitreous humor is the substance inside your eye. All right. So let's see here. So vitreous humor. Vitreous humor is protected. It's protected and has a protected environment inside the eye. So therefore, it will be less subject to contamination and bacterial decomposition. Okay, so you will understand that based on this, vitreous humor is very protected inside your eyeballs. Okay, inside your eyeballs, there is a vitreous humor. Alright, so vitreous humor ni is very protected by a layer in your eye, which is protect it from the decomposition by bacteria and also protect it from contamination. Alright, useful for the analysis of several psychoactive substances such as ethanol and biochemistry analysis. Okay, it is easy to collect and remains clear and sterile for up to 3 days or so after death. It becomes cloudy and brown with decomposition. So usually, in terms of if you want to test for ethanol, for example, then vitreous humor might be able to provide those kind of specimen up to 3 days lah. Alright. So because after 3 days, it will follow the decomposition process. Okay. Vitreous drug concentration inside vitreous humor often reflect the circulating blood concentration 1 to 2 hours prior to death, right? And that any drug found in the blood will be detected in the corresponding vitreous specimen. As a result, it may be used to distinguish ante-mortem alcohol ingestion from post-mortem alcohol formation, and may provide the only opportunity to establish an ante-mortem ethanol concentration in embalmed bodies. Okay. All right. So let's move on to the fifth specimen type, which is the gastric content. Okay. Oral ingestion is still the major route of drug administration for prescribed drugs and therefore a major compartment for the investigation of a potential poisoning. So the gastric content, as we it goes without saying lah, as we ingest something, so everything will go into your stomach, which is containing the gastric juice, right? So this is one of the primary sources of when you want to test for potential poisoning. Okay, drug overdoses, whether by accident or by intent, may readily be discovered through the analysis of the gastric content. In many cases, undissolved capsules or tablets may be discovered, which may be useful for identification. Since the gastric content is not a homogeneous specimen, ideally the entire specimen should be submitted to the toxicology laboratory for mixing before aliquoting. Right? So in this case, for example, even in blood, when you have the blood specimen, you need to be able to get the plasma. Alright, so this requires you to get the whole blood, the 10 to 20 mils of blood that you've got, to the lab, so that the lab will be able to separate the red blood cells and all the cells inside the blood and just get the plasma for analysis. even in terms of gastric content. It would not be homogeneous. It will not just be a clear liquid. So you need to get everything to the lab. Okay. All right. Specimen type tissues. Okay, so tissue commonly collected would include liver, kidney, brain, lung, and spleen. All right. So a large amount of data for drug findings in tissue exists primarily for liver and kidney. So as I mentioned, liver is the site of metabolism, kidney is the site for excretion. So these two sites might be the most important site for tissue specimen. Okay. To a lesser degree, maybe brain or lung uh, can be sampled as well. Okay. Comparison is most often between heart blood findings and those in the liver, for which the greatest amount of reference data is available. Alright, so that's what I mentioned before as well, that the blood from the heart is going to be different from the blood from the liver, because liver is a site of metabolism. So sometimes the drug might be present in terms of its metabolites rather than the whole drug lah, the whole drug structure. Okay, where the concentration of basic drug in blood is high and the ratio of liver to blood drug concentration exceed 10, a drug fatality is strongly suggested. Alright, so whenever that the concentration of basic drug in blood is high and the ratio of liver to blood is more than 10, then we would see that a drug fatality has caused the death. Okay, has not caused, maybe contributed to the death. Alright, okay. Smaller ratios tend to suggest a greater potential for post-mortem redistribution of drugs into the blood. Okay, spleen, which is an organ rich in blood, is useful for the analysis of compounds that bind to hemoglobin, such as carbon monoxide or cyanide. Okay, for some compounds, it would bind very strongly to hemoglobin. So spleen might be the place where you would get the sample from. Okay, frequently in fire death, where extensive charring is present, spleen may be the only useful specimen available to perform these assays. Right. Okay. All right. Lung and tissue. Right. Lung and brain. So lung tissue is particularly useful in cases where the inhalation of volatile substances such as solvents or freon is suspected. Okay. So lung also would be an indication, even though in terms of the specimen hierarchy in terms of tissues, lung might be at the bottom of the list, but sometimes lungs also being used as one of the primary in terms of arson cases lah. Okay, so because that you for example, when you see that when you arrive at a crime scene where the victim has died due to arson. Okay, so you are suspected that, oh, maybe the victim has died prior to it. So you need to be able to see that from the lung specimen whether the person has died prior to the arson or died due to the arson. So this is important. So you need to be able to deduce, or you need to be able to think on how to use all the information that you've got in this chapter to be able to deduce that kind of information. Okay. All right. Lastly is the brain. Brain, due to its high fat content, tends to accumulate lipophilic substances lah, such as chlorinated hydrocarbons or other organic volatiles. Right? Okay. Specimen type. The second last one is hair. So hair and fingernails are the specimens of choice in determining chronic heavy metals poisoning, such as arsenic, mercury, and lead. So this is also a site where these heavy metals have been accumulated. Okay, so heavy metals bind to the sulfuridyl groups of the cysteine molecules to form a covalent complex. Okay, all right. So keratin found in large amounts of hair and nails is an excellent source of cysteine. Right? So the interpretation of positive findings can be augmented by segmentation of the hair strands to assist in the determining the time of exposure. So hair kan there is a lot of segments from the bottom to the towards the other end of the hair. So usually when you have segmented the hair and you analyze each of the segment, you would see that how long has it been accumulated, because hair is keeps on growing kan. So if it is just accumulated at the bulb part of the hair, then you would see that, oh, maybe the exposure less is lesser than if you have detected it at a much more higher end of the hair. Okay. The last specimen, if I'm not mistaken, so the last specimen for postmortem toxicology is larvae. Okay, in cases of suspected poisoning where decomposition prevents traditional specimens from being obtained, traditional specimens here is the blood, organ, and everything. Homogenized fly larvae, usually a Californian genus, blowfly, can be useful as an alternative specimen in which drug or poison may be identified. Okay, so basically the larvae, okay, when the decomposition process has started, so the blowfly would come and infest the decomposed, okay, and then you would use this larvae, okay, because this larvae would feed on the the decomposed body. Okay, so the decomposed body, whatever drugs or poison that present inside the decomposed body would be transferred to the larvae. So you can use this larvae as an alternative specimen to the traditional specimen, which is what we have discussed before. All right. So this transfer of poison from the decomposed body onto the larvae, you would expect that the drug or the poison would have been metabolized, okay, because the drug has been metabolized inside the decomposed body prior to the death lah. So the drug has been decomposed, and it is going to be further metabolized inside the larvae itself, because the larvae also has its own digestive system. Okay, so you'll be able to provide the useful in diagnosing the cause of death. Okay, but since these substances are not drugs, abuse and therapeutic drugs can be used, but common poisons are used for committing suicide. Okay, so you need to be able to deduce that whether there is a drug present inside your larvae specimen. If there is no drugs present, maybe its metabolite is going to be present. Okay. All right. So this is the guide to the collection routines toxicology specimens. You can have a read at them. Okay.